Hello everyone, my name's Kaylin and today I'll be discussing the restorative effects that serotonin can have in fear memories. So one phenomena in the memory paradigm is infantile amnesia. This describes a process during infancy where early memories are rapidly forgotten. Infants and adults form and express memories very differently. Adults have a stronger memory consolidation than infants. Memory consolidation is the process by which recent experiences are transformed into long-term memory. Infants who are healthy take longer to consolidate memories during their early years and experience infantile amnesia. It's very important to assess the impact that early life stress can have on adults. The long-lasting influence and the paradox of infantile amnesia is mentioned in the Burchest Early Intervention Program. This is a very well-studied example of the impact that adverse early life experiences can have on development. Children exhibited significant differences in brain activity, and this study demonstrated the severe neglect and institutionalization during the first 4 to 5 years of life, which resulted in deficits and delays in cognitive and socio-emotional behaviors. Attachment, emotional responsiveness, and IQ are severely compromised in these children who have been institutionalized and receive lack of care. Neglect can produce severe physiopathologies such as depression and even schizophrenia. Results found that the earlier these children were removed from this institution and transformed to foster care, the better the outcomes are for development. There's a sensitive time period during which early intervention can repair the effects of childhood adversity. So now that we know what impacts early life stress can have on our overall development, let's have a look at what exactly happens to our memories when we experience early life stress. When infants are exposed to early life stress, they exhibit an acceleration in memory development. This means that infants can have an adult-like presentation of memories. It means that their fear memory can become very consolidated and very strong, and they don't exhibit the typical infantile amnesia that most healthy infants have. Instead, they develop a type of fear that's very resistant to extinction training and has become very conditioned very quickly. Over the last decade, the gut-to-brain axis has gained traction as a topic of interest within the scientific and public spheres alike. In the gut, there are trillions of microbes. This is known as microbiota. In previous studies, it's been seen in animals that the brain is affected in the absence of microbes in the gut. Specific strains of bacteria has had an impact on animal behavior, and this has been translated to humans as well. It's been demonstrated that the GI tract influences brain function. This happens throughout many different systems. However, one key neuronal component is the vagus nerve, which is shown on this diagram. The vagus nerve is one of the connections between the brain and the gut. Specifically, the vagus nerve sends signals to the gut and the brain, and it's very complex. However, the most common way it acts on the brain is through the bloodstream. Signaling from the gut microbiota interacts with the central nervous system by regulating brain chemistry and influencing the endocrine systems associated with stress response and memory function. Many of these effects seem to be strain-specific, meaning that specific microbes in the gut act on specific parts of the brain. One particular study that took my interest was the role of the gut microbe called bifidobacterium had on reversing this adult-like fear expression in rats, and the early experiences that stress has on this abnormal memory that the infants have. So, as we know, exposure to early life stress is a powerful risk factor for many physical and mental outcomes. Early life stress is associated with changes in the gut microbiota and has been shown to modify the microbiota composition in many species across rodents and monkeys. Previous research using this model has documented the reversal of this premature maturation of fear memory through probiotic treatment. Due to this finding that mental memory deficiencies may be reversed via early life colonization with bifidobacterium. This is a type of probiotic within the gut. And looking at this study, it's highlighted gaps in this area of research. no study to date has looked at the precise mechanism by which the restorative effect of probiotics can have on fear memories in infants. Looking at this gap in research, it's led me to believe that serotonin may play a role in mitigating the effects that the probiotics have on restoring the age-appropriate developmental trajectory of fear memories. This is because the gastrointestinal tract has 95% of serotonin, which is a key neurotransmitter that influences behavior and neurodegenerative disorders. Animal research models suggest pathways of communication along the gut-to-brain axis, especially in the production of neurotransmitters. Thirdly, the amygdala is the most important brain region for processing fear, which is modulated by serotonin. Investigating the role of serotonin in maternally separated rats is important for 3 reasons. Firstly, it can extend on treatment options for anxiety and depression by highlighting the importance of the brain-to-gut microbiota for memory development. Secondly, it can provide causal mechanisms for specific neural circuits underpinning fear expression and extinction in infant rats. Thirdly, the reversal effects of probiotics on maternal separation is a largely understudied area. Closer examination on the mitigating effects of probiotics on memory restoration can provide clinical treatments for childhood adversity and prevent long-lasting effects of fear memory from this adversity. Therefore, the aim of this study is to investigate the possible role that serotonin has on restoring the normal developmental trajectory in stressed rats. So I'll be using rats that have been maternally separated. So rats that have been separated from their mother on the first day of birth. This induces a stressful environment that makes the rats have a longer lasting fear memory that's resistant to extinction. So it's hypothesized that the rats in the maternally separated group treated with the probiotics and PCPA, which is a serotonin antagonist, meaning it blocks serotonin production, will exhibit greater freezing responses the day after extinction. This is because serotonin is blocked and it may be the causal mechanism underlying the restoration of age-appropriate memory development. So I'll be using 2 groups. The first group is maternally separated rats, and the second group is standard reared, which means rats that have not been separated. There'll be 4 conditions. The first condition in the maternally separated rats is the probiotic Bifidobacterium infantis and the serotonin antagonist. Second group, standard reared rats, will be injected with also Bifidobacterium infantis and the serotonin agonist. And the maternally separated rats will be injected with a vehicle treatment, which is saline, and so will the standard reared rats. So maternal separation will be induced by separating the pups from their mother during P2 to 14. So that means during postnatal day 2 to 14. For 3 hours at randomized time points during a 24-hour time period. The standard reared group will remain with their mothers. So following this period of maternal separation or no maternal separation, rats will be injected with either PCPA, which is a serotonin antagonist, or serotonin directly into the amygdala. Rats will also receive probiotic treatment. The probiotics will be mixed into the rat's drinking water. Okay, so now phase 3 begins. Phase 3 will include fear conditioning, extinction training, and then testing of that extinguished fear response. So 24 hours after the injection of the probiotic treatment, rats will receive 14 presentations of white noise CS and tail shock US, so unconditioned stimulus and conditioned stimulus pairings. Extinction training will commence, and rats will be presented with the white noise but no shock, which extinguishes until we see a reduction in that fear response, which is the freezing. Rats will then be tested on their retention of fear. The white noise CS will be presented, and the rats will be scored for their level of freezing, which indicates their fear memory that they have to that conditioned stimulus. So if we look to the left of the slide, the percent of freezing to the tone for maternally separated and standard reared rats receiving that probiotic treatment, as can be seen, there's no clinical difference between freezing rates at test. This is because serotonin has not been blocked and the probiotics aid the extinction training and also have an effect on restoring that developmental trajectory of the fear memory. So if we look to the right-hand side, the maternally separated rats, that's injected with that serotonin antagonist, and the probiotics have a higher freezing rate at test, which indicates a stronger fear memory, which is also resistant to extinction training. This suggests that fear memory expression in maternally separated rats following the probiotic intervention could be mediated by the changes in serotonin production.